I'm Daniel Levine, and this is the Bio Report. One of the challenges in treating the neurodegenerative condition Alzheimer's disease is intervening early enough in the course of the illness to provide meaningful benefit. AltPep is developing therapeutics with companion diagnostics that target toxic alpha sheet containing oligomers, which are thought to form very early in the disease and act as molecular triggers of downstream amyloid pathology. These structures are believed to represent some of the earliest detectable stages of Alzheimer's and promote the formation of amyloid plaques, a hallmark feature of the condition. We spoke with Valerie Daggett, founder and CEO of AltPep, about the relationship between alpha sheet oligomers and the onset of Alzheimer's disease, how the company's synthetic peptides are designed to bind and neutralize these pathogenic agents, and the potential for this platform to be extended to a broader set of amyloid diseases. Valerie, thanks for joining us.
Well, thank you for having me. It's a pleasure to be here.
We're going to talk about neurodegenerative conditions, Alzheimer's disease, and gaps in treatment and diagnosis for these conditions. AltPep is developing synthetic peptides to both diagnose and treat these conditions. A lot of focus to date in Alzheimer's has been on amyloid beta peptides that accumulate in the brain, but therapeutic strategies targeting amyloid beta have not been terribly effective as a means to treat the disease. Why is that?
Well, mostly you're waiting until there are symptoms to begin treatment. And the treatments out there at this time, the FDA-approved treatments target plaques, mature plaques. And we know now that the disease begins 10 to 20, some people even estimate 30 years prior to that, prior to the onset of symptoms and being able to detect the plaques, say with PET imaging. And so it's really important that we have ways to detect the disease earlier so that we can treat earlier.
Is there any way to quantify both the unmet need for earlier diagnostics as well as treatments?
Um, that's a great question, but it's a hard one. There are a lot of different numbers out there. I'll throw out a number. I think we're probably dealing with at least 500 million people worldwide that have this disease, some form of the stage of this disease, and so it really is a continuum. Think of it from people incubating the disease to having full-on dementia of the Alzheimer's type. And we're really, in terms of unmet need, we're missing that critical period when you could get in earlier, and we just don't have firm numbers on that, because there haven't been diagnostics that could detect it. And so we're losing out on critical time when we could intervene and maybe do something that's truly disease modifying, or at least hold it at bay before moving into more advanced stages of the disease.
You're focusing on alpha sheet toxic oligomers and the role they play in Alzheimer's disease and other neurodegenerative conditions. Can you explain what those are?
Yeah, so it's been known for a while that there are toxic oligomers of the amyloid beta peptide that precede plaque formation. And so my lab, we started studying this initially computationally because it wasn't possible to characterize the process experimentally. And we found that amyloidogenic proteins were passing through a state that normal proteins did not, and that these it this structure was a new structure that we discovered. And so this was set against a lot of work going on in the field where people were looking at oligomers, but they couldn't characterize them. So they were known to cause a variety of effects. And then we took the approach of characterizing the process in detail and then being able to hypothesize that this structure that's going on in the animal studies and in humans is related to what we had discovered in the computer through realistic computer simulations. And so that's sort of how it started. It started as a hypothesis that alpha sheet structure is the structure of the toxic oligomers people had been seeing.
This was work you had done at the University of Washington. Has that progressed now? Have people come to accept that the role of these alpha sheet toxic oligomers in the development of these diseases?
It's so I've been at it for about 30 years, but, and it's certainly people are more receptive to the idea now, and there are many labs that look at this. We're not the only ones. I mean, we have our own special space in this, what we're doing, but, I think it's being more and more accepted, but you still, in terms of what you're seeing in the clinic, you're looking at things that have been there a couple of decades, and so it takes a while before you see it play out in the commercial sector, say.
At what point did you realize there was a basis for a company in what you were doing?
So, um So after we discovered this new structure and we really put it through its paces because we said nobody discovers a new protein structure, a new structure of the main chain of the protein, and we kept seeing it over and over in amyloid proteins, but not in normal proteins. At that point, we said, well, we have something that potentially here could be a game changer because it could allow us to come into the process much earlier than going after plaques or protofibrils or later stage aggregates. And so we focused on that, and in fact, I made a lateral move at the University of Washington. So we've been supported by NIH. This was in the basic research end of things at this point, and then moved to a bioengineering department where I got a wet lab, and we were able to really study this in detail. And it took about 15 years and a lot of biophysical studies to demonstrate that amyloid beta peptide, for example, does form the structure we discovered, alpha sheet, and it's in the toxic oligomers, and it's highly correlated with toxicity. And so after, kind of pushing this hypothesis and seeing that we were getting from many different angles proof that this was going on in not just Alzheimer's, but other amyloid diseases, we said, well, now's the time we have to think about taking this out. It's not going to go anywhere. We're not going to actually be able to help anyone if we can't get it out of the university. And that started the process, the whole intellectual property process, which is kind of its own story because we patented the structure, not the sequence. And the patent office works on the basis of composition of matter. And so that was a long process of back and forth, 8, 10 years to get the IP. And at that point, we were ready to spin out, but we couldn't do it before the IP was in place.
And you're focused on developing synthetic peptides to go after the alpha sheet oligomers. Why use synthetic peptides rather than antibodies or small molecules?
Yeah, so, the reason for that is, because we're targeting structure, not a surface effect of some side chains. So antibodies work by binding basically 3 to 5 amino acids on the surface of a protein, and that can be in the case of with these amyloid diseases, you've got something that starts out in one structure, it adopts, one or more other structures and different aggregate forms. And the antibodies are going to bind to that epitope, that surface, in any of those different forms if it's exposed. And so the antibodies have been largely they're nonspecific in a sense. They're highly specific for a sequence, but not for a structure or a particular Well, in this case, for the toxic oligomers. And so if they bind toxic oligomers, they also bind the other forms of the amyloid beta peptides. So they can't distinguish between, they can't be used for detection, for example. So with the peptides, we're going after the structure. So we discovered this structure, alpha sheet, and the peptides also have alpha sheet structure, and it's complementary and it's highly selective. So you've got, imagine toxic oligomers moving around your brain and they have alpha sheet. And then we have our synthetic peptides that have alpha sheet. They bind very tightly to that, so they can serve as both a biomarker and for detection, as well as that tight binding for using them as therapeutics. And you have to have, as opposed to using a small molecule, for example, small molecules tend to bind into active sites or cavities. It's a surface that you need to bind to in the toxic oligomers. And so by using the peptides, we get highly selective binding through the alpha sheet structure. Excuse me. And we've got enough of a surface area there to give us nanomolar binding.
AltPep is developing both a blood-based diagnostic soluble oligomer binding assay, or SOBA, and a soluble oligomer binding inhibitor, or SOBIN AD. Let's start with the diagnostic. How does it work?
So it's all this complementary binding between the toxic oligomers, the alpha sheet structure, and the alpha sheet structure in the compound. And the reason is it's got a molecular dipole, and you've got these charge interactions that bring them together. And it's distinct from the other forms of A beta that are floating around. And when what we did for the technology for the detection is we created a plate-based assay, and instead of using an antibody as a capture agent to pull down where you'd apply your cerebrospinal fluid or your blood, we use our peptide. And so we're then pulling in on the basis of structure, the things we capture on the plate. So I like to think of it as kind of a battery analogy. So you've got, if you've got them aligned right, you've got a strong interaction. And if you don't, you're not going to have the binding. And so we get that binding to the surface in the plate, and we also develop some more technology around that to get so that it's highly sensitive. So you have a peptide on the plate to pull down toxic oligomers. And then those toxic oligomers could be from the amyloid beta peptide, or it could be from alpha synuclein, or one of the other 50 amyloid diseases, the proteins or peptides involved in these other amyloid diseases. So then we use conventional antibodies, like I was talking about, where you're just looking at an epitope on the surface to tell us the chemical entity, what it is we've captured. And then we know which disease we're dealing with.
And where are you in development of that?
So we are very, very close with our LDT, our lab developed test. We've finished up the validation of it and doing the paperwork, and so we'll be ready to go with that. Crossing my fingers, hopefully in the next week or two. And then we also have FDA breakthrough status moving forward to the IVD. So what we're doing is the LDT first, and we're looking at, for example, a number of different samples from pharma, from clinical trials, things like that, and then with an eye to moving towards the FDA validated test as well.
So just to be clear for listeners, you say the next week or two, so before the end of 2025.
That's the goal. Yep.
And what is Sobin AD and how does that work? This is your inhibitor.
Right. So that's the soluble oligomer binding inhibitors. They work on the same basis as the detection, the binding of the alpha sheet in the toxic oligomer to the binding in the peptide, not necessarily the same peptide, and in fact, it's not for our AD or Alzheimer's therapeutic that we're looking at. And so in this case, though, of course, it's not immobilized. What we're using is intranasal administration to the brain. And then these toxic oligomers, unlike plaques, are diffusing around the brain. Our compounds bind, they bind very tightly. and then they stimulate clearance via the brain immune cells, the microglia, and they get so that the toxic oligomers get carried away and processed by the microglia. We stimulate that process.
And where are you in development on that?
So we are in our getting our IND together and or the equivalent in Australia, and we're looking to get into the clinic next year.
And what's known about it from the preclinical studies you've done?
So we've done a variety of animal studies and in vitro studies and such, and our compound is um, doing. We did, for example, one thing to show since you've got toxic oligomers, and it's a novel, novel structure going after a different mechanism, all of that. We want to show that the toxic oligomers are actually causative. So we made pure versions of these alpha sheet toxic oligomers. We injected them into the brains of mice, wild type, naive, non-disease mice, and we were able to induce symptoms of Alzheimer's. And it parallels known effects and behavioral effects, cognitive effects that you see in transgenic mice. And then we gave intranasal delivery with our compound, our Sobin, and we were able to prevent and, in fact, even reverse those effects. And from there, we moved on to the more widely used transgenic mouse models. And now you've got a different situation there where they're constantly making toxic oligomers, as a human would be once they start making them. And we did a couple of different studies where we went in at different stages to see what effect we would have. So we went into animals kind of at the mild cognitive impairment stage, and we were able to counteract the effects. And so we would see improvements in their learning, their memory, different behavioral hyperactivity disorders and such with our compound. And so that's getting in early before you have substantial damage and such in the brain. So then we did another study where we started with younger mice and we carried it out for a year of dosing and took it all the way into the plaque stage so that we could see the effect. So the idea is these toxic oligomers are the trigger for the molecular pathology, and everything else is downstream. And so one thing you'd look for is you'd say if your compound is working and you're binding these toxic oligomers and they're being cleared, you should have less plaque formation. And so we checked that after when they were 15 months old, after a year of dosing, and that was indeed the case. So we're not binding to plaques, we do not bind to plaques, but we prevent them. And then more importantly, we neutralize those toxic species that are upstream so that they aren't doing damage and signaling isn't impaired and the consequences of that, learning and memory. So we've done a lot more, but the gist of it is it's working quite well in animals.
So you mentioned earlier in this conversation about the importance of early diagnosis and having success in treating the condition. How early might your diagnostic be able to identify someone developing the disease, and how late in treatment might the therapeutic have benefit?
Um, yeah, so good questions, and it's a little hard to give, really direct answers until we do it. But we've done studies where we've used retrospective samples, banked samples where there's been longitudinal follow up, or we've asked for follow up based on predictions we've made of preclinical cases, and we've been able to validate those. And it's dependent in terms of answering your question and putting it on a timeline. It depends on when those samples are banked. And our record so far is catching it 16 years ahead of time. And but it again, it depends when they're banked, and then when they're followed. And so we've gotten a lot more samples now where we're looking at that in more detail to get a feel for it. But in principle, we pick it up when they start making these toxic oligomers. So whenever that starts happening, we can see it, and then we just need validation that, in fact, they move on. In terms of going into the clinic, we intend to go into early disease initially, where mild cognitive impairment, and then we will move forward in time. I was surprised, in fact, that we saw such a robust effect in animals that already had disease. In humans, if they're advanced, I think we would need combination therapy. I don't think we can't reverse 20 years of damage, for example.
And is the expectation that SOBA would be used as a companion diagnostic with SOBIN?
Yeah, so, SOBA is really important for our therapeutic program because, what's really nice, and you rarely have this, in this field, is a biomarker that's truly linked to your target and your mechanism of action. So, by detecting SOBA, it allows us to know that our patient has our target, and it allows us to hopefully bring in people who are in the incubation stage and don't yet have symptoms as well, and identify those and stratify patients. So, but also importantly, SOBA then, the idea is we're going to be taking blood samples and we can track the toxic oligomer burden with therapy. So we've done this with in our animal studies, and we can see that intranasal delivery of our Sobin leads to a drop in toxic oligomer burden as measured by SOBA. So, as you would for like a tumor burden for cancer.
You're also pursuing a SOBA and SOBIN for Parkinson's disease. And I'm curious, is there a difference in alpha sheet toxic cellular oligomers in Alzheimer's compared to compared to Parkinson's, or are you using the same test and the same therapeutic?
Yeah, so for example, alpha-synuclein also has alpha sheet structure. And as do we've looked at 14 different amyloid systems. They all do. They all funnel through alpha sheet. We're 14 for 14 so far, and our compounds work against them. But that doesn't mean they all have different sequences, and the precise arrangement of that alpha sheet structure is going to be different. But since we're detecting on the basis of structure, they all have that structure, so we can detect them with our SOBA assay. Now, we'll see if moving forward as we move into other diseases, if they need some tweaking with the assay, and what I mean by that is longer incubation times for some than others or something like that. But fundamentally, the same assay we already know works for Parkinson's and, Alzheimer's. And you just as I mentioned, we use a a normal antibody to tell us, are you alpha-synuclein or are you amyloid beta peptide?
Given that this is a blood test, how do you expect it to be used? Would it be on people with family histories? Would it be on people who are showing early signs of developing diseases potentially? Or do you see it as a broad blood test if you're past a certain age?
Yeah, so I'd see it as both that you mentioned. I mean, and I get a lot of emails from people who would like to use it because they have Alzheimer's in the family, or they're starting to lose their keys and things, and just wondering wanting to see if they have toxic oligomers. But, aside from that, this is kind of the moonshot. It's not going to happen, soon, but we'd like to see it as a screening assay, like a cholesterol test where somebody's getting it every year, every other year, and you catch it early and begin immediate treatment. And that, might be with our therapeutic, ideally, or with others. And so I think there are many ways this could be applied. Now, one thing that from the very beginning would be helpful is there's so much comorbidity in this area of these neurodegenerative diseases, and they're kind of overlapping symptoms in the early stages. And so even a test that could definitively say, no, you don't have Alzheimer's, is of value because then you can start to look for other causes of the cognitive impairment, for example.
you've been able to raise significant capital in what's been a challenging time for early stage biotechs. Why do you think you've been successful, and what's the discussion with investors like these days?
Um, I think it takes, well, special investors who are willing to invest in something that's very different from what everybody else is doing. And so it's yeah, we're fortunate in having some good backing. And then as far as talking with investors now, of course, things have, continued to be, problematic with the market and such, and the goalposts keep changing. And so, it's been we still have, support, and of course, we're gonna need more money in the future, but we're moving into the clinic, and now, people and investors and others, they really want some clinical data and to show that it's safe in humans and that sort of thing, and so, that's what we're focusing on at this point.
Eli Lilly is among your investors. Do you see turning to a big pharma partner for late stage development and commercialization ultimately?
Yes, especially the stage we're at now, I think our real strength is the science, and there's so many of these amyloid diseases, and as I mentioned, we've, worked on many of them in my academic lab at the University of Washington, and so in a way, I view it as kind of a conveyor belt. We can do the basic research, and then as we move into the clinic with one disease, Alzheimer's, we're already working on Parkinson's, that then we put more emphasis on that, and then we bring in the next disease. And then, but I don't this may change over time, but, I don't see commercialization and a sales force and all that on the horizon for us because I'd really rather focus on, these diseases and getting them out there and getting them in the hands of people who can commercialize them well, and then, like I said, move on to the next disease.
Beyond Alzheimer's and Parkinson's, are there other amyloid diseases you think would be good candidates for your approach? And would it extend to amyloid diseases outside of the brain?
Yes and yes. A couple that we're quite interested in and have done quite a bit of work on are, systemic amyloidosis associated with transthyretin and heart failure, and then also, type 2 diabetes, which is the biggest amyloid disease in terms of numbers. And then we've looked at various others, including, bacterial, effects, with that's kind of another story, but there are diseases associated with bacteria that where amyloid inhibition is can be quite helpful.
Valerie Daggett, founder and CEO of AltPep. Valerie, thanks so much for your time today.
Thank you.
Thanks for listening. The Bio Report is a production of the Levine Media Group. To automatically download this podcast each week, subscribe to our RSS feed or through iTunes or other podcast manager. To join our mailing list, go to levinemediagroup.com. We'd love to hear from you. If you want to drop us a line or are interested in sponsoring this podcast, send an email to danny@levinemediagroup.com. Special thanks to Jonah Levine, who composed our theme music, and the Jonah Levine Collective, which performs it.